Okay, so we haven't done one of these in a while. This is chapter 10, photosynthesis. We're gonna start hitting this on Wednesday, which will be tomorrow. Some lab work and kind of some background information. So we just finished up respiration. Obviously, difficult topic. Photosynthesis literally is the reverse of photosynthesis. So if you think of respiration, so if you think of respiration being glycolysis, the Krebs cycle electron transport chain, if you turn that around, you'd have electron transport chain, Krebs cycle, glycolysis. That's pretty much how photosynthesis works. They don't use the same terminology. Instead, they call it the light reaction, which is an electron transport chain, the Calvin cycle, which is sometimes referred to as the dark reaction. And then from there, G3P is made, which then can be turned into anything that plants need, not only just glucose. So, we talk about this term that life on Earth is solar powered, meaning that whether you directly eat plants or you eat the things that eat the plants, all of our energy on the planet is coming from photosynthesis. There are other types of synthesis as well besides photosynthesis. There's something called chemosynthesis, which we kind of briefly touched on. It actually happens to do with chemical synthesis. So instead of using photons to drive the synthesis, it uses chemicals to drive the synthesis. This used to be super common on the planet until we developed an atmosphere and until it became a little bit more less harsh for organisms to live on the surface of the planet. We still see chemosynthesis around places like volcanic vents. We see it deep in the ocean around hot spots in the deep parts of the ocean. So the players are really simple: autotrophs and heterotrophs. Autotrophs are things that make their own food. They're gonna be the process of being autotrophic is going to be an anabolic reaction where you're taking CO2 from the atmosphere and you're turning that CO2 into materials that we can eat, such as glucose, proteins, lipids, carbohydrates. Heterotrophs, on the other hand, are going to use catabolic reactions to take those things and then turn the sugars, fats, lipids, proteins, and turn those things into smaller molecules, such as CO2 and water, to extract the energy from these things. So we can talk about photo versus chemoautotrophs. This is what we were talking about: photosynthesis versus chemosynthesis. So photosynthesis obviously uses the energy found in light, and that energy in the light drives the reactions that take place. Here are just some examples: obviously plants, algae, protists, prokaryotes. Chemoautotrophs harvest energy from using inorganic substances such as sulfur and ammonia. Those would be the chemicals that are found there. We'll talk a little bit more about We'll talk way more about photoautotrophs than we do chemo. The process is similar, but for the most part, that's where we'll spend most of our time. So obviously, heterotrophs fall in this category of they can hetero, meaning other, trophic, where you eat. These would be things that cannot make their own food. So obviously plants and algae here. These are going to be all of our animals. Then we can even break those animals down into smaller categories such as herbivores, carnivores, omnivores, and decomposers. Obviously, herb eat veg, carn eat meat. Omnivores are us; we eat a little bit of both. And decomposers don't eat veg or meat. They take the dead organisms and then turn those back into products that can be used, reactants that can be used for photosynthesis and chemosynthesis. So chloroplasts, we'll actually draw some pictures about this. We'll talk about how they work, what they do. We'll talk about the pigments found inside. There's chlorophyll A, chlorophyll B, which are greens, and then we'll talk about xanthophylls, which are yellow, and so on and so forth. Carotenoids, which are orange. So we'll actually draw some anatomy of a leaf. So leaves are, for the most part, are thin and wide. Why are they thin and wide? By being thin, they can be lightweight. Why are they wide? It gives them more surface area. And the idea is that if you look here, here's a typical leaf, and what they've done is they've taken a cross section or cut through the leaf, and they show you the different parts of the leaf. Ironically, when you're looking at leaves on a tree, let's say, you would see that one side is shiny, one side is dull. The shiny side is the top side of the leaf and has this waxy coating on the outside, and the bottom side of the leaf is kind of dull. I remember my dad used to make the comment all the time that we can see when you can see the bottoms of the leaves when the wind blows, you know it's going to rain. Which I don't know if it's true or not, but who knows? Um, anyway, so we'll talk about anytime you see PHL or the PHYL, we're talking about plants in some respects, like chlorophyll, mesophyll. So it's a plant reference, just kind of a root word for us. We'll do this thing in class where we're going to do a stomata peel, which is where you take nail polish and you rub it on the bottom of the leaf and you peel. You peel that nail polish off with some clear tape, and it allows you to see these small openings in the bottom of the leaves called stomata. Okay. Chloroplasts are structures that have their own DNA, very similar to mitochondria. So these are another one of these items that have been pulled into cells through endosymbiosis or endocytosis, where they were swallowed and never metabolized, and then still live in plants today. So we can make some evolutionary, I guess, ideas or guesses here. For example, mitochondria are found in all eukaryotic cells. So we can make the assumption that some common ancestor to both plants, animals, fungus, all eukaryotic cells, absorbed, swallowed, engulfed a bacteria that became mitochondria, and then those mitochondria then showed up in all subsequent cells after. At some point along the line, plants went a different way. Plants or even algae went a different way. Is they swallowed some type of thing, in this case, chloroplast, which would have been some type of bacteria that is able to be photosynthetic, and obviously that kind of divided plants and animals at that point. So we'll draw a picture of this. Obviously, we'll talk about the different parts. We'll talk about where things happen. Super important to know kind of where things are going on inside the cell. So chloroplasts, they have three membranes. They have an outer membrane, an inner membrane, and the innermost membrane called a thylakoid membrane. If we go back to this picture here, you can see there's this outer membrane, there's a secondary inner membrane, and there are these green discs or stacks here. So this is the first membrane, second membrane, third membrane. This third membrane is where all the action happens. This is where the pigments or the colors in the plants are found. And these pigments then are excited, or they're most active when there's light, and we'll talk about how that action actually happens. So basic formula: 6 CO2, 6 water, some sunlight. Arrow there, so we know this is in the presence of some type of enzymes. There's a series of enzymes that then will drive the process of making C6H12O6, glucose plus 6 oxygen molecules. And these oxygen molecules can then be released into the atmosphere as waste, or they can be used inside the plant for them to do respiration. Once again, we'll talk a little bit more about this as we go in class. So this is a guy, C.B. Van Nile, and he was the one that discovered where the different portions of, or where the reactants go for photosynthesis. One of the ways you can do this is you can tag molecules. So, for example, you can use an isotope of carbon, okay, or an isotope of oxygen, or an isotope of hydrogen, and you can feed those to plants. So you can use, carbon 14 as opposed to carbon 12, which is the normal carbon for CO2. You can use hydrogen that's got a neutron in it, so it's a little heavier. You can use an oxygen. Oxygen 17, oxygen 15, some isotope of oxygen, and then you can track where those things show up in the products in the plants. And when he did this, what he found was that obviously the carbon that's found in the CO2 goes directly to sugar. The oxygen portion of it goes to the sugar, a portion of it goes to the water molecule, and that the hydrogens that are found in in water directly the hydrogens directly go here to water, so just water to water. But the oxygen produced from plants is directly derived from water. So no water source, no oxygen. Where this becomes important is that there are multiple types of photosynthesis. Where in respiration there's only one type. There's actually C3, C4, and CAM plants. So 3 major types of photosynthesis. It depends on where they live, and it depends on how much water that is available. So plants that have plenty of water available do C3. Plants that have limited access to water sometimes will do C4. Plants that have no water will do something called CAM, which we'll get into a little bit as we go. So excuse me. So the two major parts of photosynthesis, obviously, the light and dark reactions. The light reaction drives dark reaction. The light reaction, they call it that because it only can happen when there's light energy available, whether it's from the sun, whether it's through artificial light sources, but it can only happen then. The Calvin cycle is named after a guy, oddly enough, named Melvin Calvin. His parents must have hated him. Anyway, Calvin discovered the process of this of the cycle that produces organic molecules from CO2. So in the first reaction, think of this as like the electron transport chain. The light reaction is taking light energy, converting it to chemical energy, which in the case that the only one we can use is ATP. So it's going to convert light into ATP. The Calvin cycle then uses that ATP to then smash CO2 and water together to get organic compounds. So light reaction, we talked about Uber and Lyft. Here there's only Uber. It's got a little different name. If you remember NAD from respiration, NAD was an electron carrier. It carries hydrogens places, carries them specifically to the electron transport chain. In the case of in the case of the light reaction here, what's going to happen is NADP for plants, really means phosphate, but NADP for plants is a good way to remember it, carries hydrogens from the light reaction to the Calvin cycle. And those hydrogens are going to be incorporated in the new organic compounds that are made. Remember, hydrogens will really carry all the energy. Okay, there's this long term called photophosphorylation. It's a simple way of saying we make ATP through light. So photo, light, phosphorylation to add AP. Once again, we'll draw some diagrams and talk more about this in class. Big picture, what's going on here, we have in a plant cell, we have multiple chloroplasts. Chloroplasts tend to be in greater number in plants on the surface of the cell as opposed on the surface of the plant leaves as opposed to deep inside, which we'll talk about the anatomy there. Sunlight or light of some color is going to drive these reactions. Okay. In green plants, all colors except for green actually are used in the light reactions, and we'll get into that a little bit more. One of the reasons is because green plants are green is because they reflect green light. Kind of like this pen here is green because it reflects green light as opposed to absorbing it. So light that is absorbed is what drives these reactions. During that process, water is split. All right, when water is split, what happens is these hydrogens end up here in the light reaction, and oxygen is not used, so it's removed as a waste product. We talked about, somebody, I forget who it was, asked the question about why is oxygen always O2. Oxygen's diatomic. It's two fat guys playing tug of war, and they play well with each other, so they stay as O2 like this. The only reason you need oxygen is for your for the respiration is for the mitochondria. If we look at the second part of this, so here's our light reaction, taking light, water, making oxygen. Second part of this is going to take the CO2, kind of similar to the Krebs cycle, all right, but instead of releasing CO2, it's going to absorb CO2, and instead of spitting out CO2, it spits out sugar. So it's literally the reverse of respiration. In respiration, we'd have glycolysis over here. Sugar would come in, go in as pyruvate, CO2 would come out, NADH, ATP would be used, NAD would go to the electron transport chain. So it's really, literally just the reverse. So there's three parts to the Calvin cycle. They talk about the first of those parts is called carbon fixation. So it's a way of taking carbon from the atmosphere and adding it to molecules that already exist in the Calvin cycle. All right. So I'm going to stop here, and we'll make a second video, and hopefully this will help.